All right, are there any questions? So last time we got started with glycolysis, right? Any questions on that? So the starting point for glycolysis was basically fructose 6-phosphate. And then we went through a whole bunch of steps, right? And then somewhere down the road, right, we came up with phosphoenol pyruvate, which is abbreviated as PEP, right? And then the next step was you go from phosphoenol pyruvate to pyruvate, right? And this reaction is called the pyruvate kinase reaction. Because the phosphoenol pyruvate actually loses a phosphate, right? And ADP goes to ATP, right? And it is also called the phosphorylation that happens here, ADP going to ATP, it is also called substrate level phosphorylation. Right? And then once you go to pyruvate, all right, what happened after that? Anybody remember? Acetaldehyde and then ethanol. Okay, that's one possibility. All right. So it could either go to alcohol, acetaldehyde, and then to alcohol. All right, ultimately to alcohol, and in the process you regenerate the NAD plus that you need. Okay, that you used up, so that the process continues. So one possibility is to go to alcohol. What is the other possibility? Go, what? Lactate. Yeah, good. Lactate, yeah. So, and there is an enzyme called lactate dehydrogenase, and each of these, it has got the potential to not the potential, it actually regenerates the NAD plus, so it keeps glycolysis going. Otherwise, all the NAD plus will get used up, right? And you're not going to be able to continue the procedure. Okay, because earlier on you did need the NAD plus to go from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, right? If you look further up. I'm not asking you to memorize all the steps, all right, but the important ones you should know. Now this stuff is actually does not use any oxygen. It is anaerobic, all right? It's anaerobic, all right? So if there is no oxygen in the environment, then the glycolysis will keep on running. It's extremely inefficient, right? Because it produces only 4 molecules of ATP per molecule of sucrose, right? But the good thing is that along the way, right, because you're not oxidizing glucose or fructose completely to carbon dioxide and water, right? So you are producing intermediates that are needed for biosynthesis, right? So now if you have cells in the absence of oxygen, they will run only glycolysis, right? So so normal cells, you know, if oxygen is a limiting factor, they will try to get their energy by running glycolysis, right? But and this procedure, right, or this fact is known as the Pasteur effect, right? So let me just mention that. This is called the Pasteur effect, that the normal cells, named after Louis Pasteur, right, who first observed that, right? Now, if it's a cancer cell, right, then even when you have lots of oxygen, so normal cells, when you have lots of oxygen, they will switch from glycolysis to other processes that are more efficient. But cancer cells, even when you have a lot of oxygen, right, they will still continue to run glycolysis. And there are lots of reasons for that. One reason is that those intermediates are needed. So you don't want to oxidize everything to carbon dioxide because this is a rapidly dividing cell, right? Another reason could be because the oxygen supply is limited in that environment, right? Because the tumor is growing, right? So that is called the Warburg effect. So again, I mean, this has nothing to do with plants, but it is in the context of cancer. So Warburg effect means even in the presence of oxygen, it still continues to run glycolysis, right? But whenever oxygen is a limiting factor, it is going to switch to glycolysis, the less efficient energy generation procedure, right? Now, so this completes our discussion of glycolysis, all right? So now a number of, so what happens if you do have oxygen, right? Then the pyruvate, if you have a lot of oxygen in the environment and let's say it's a normal cell, not a cancerous cell, right? Or it's a normal plant cell, then this pyruvate, right? It will go to the mitochondria, right? This stuff, this glycolysis happens in the cytosol of the cell, right? So it will go to the mitochondria, right? Where there will be another cyclical procedure that is used to generate a lot more energy, right? So it is going to go to mitochondria, right? And then I'll tell you what steps will take place after that. Now, one more thing that I should point out is that the phosphoenol pyruvate, the PEP, right, instead of going to pyruvate and then, going to alcohol or lactate, right, or even for that matter, going to mitochondria, right, pyruvate going to mitochondria to take part in the TCA cycle. Another possibility is for phosphoenol pyruvate, it gets acted upon by some other enzyme that is called PEP carboxylase, right? Right? Phosphoenol pyruvate carboxylase. Now, see these sugar. Remember, fructose and glucose. These are how many carbon sugars? 6 carbon sugars. All right. But you broke them up into 3 carbon sugars. All right. Even this phosphoenol pyruvate also has got 3 carbons. Right. So if you carboxylase means it's going to add more carbon to it. Right. So PEP carboxylase will convert phosphoenol pyruvate into a molecule that is called oxaloacetate, and we will abbreviate it as OAA, right? And you don't have to memorize the detailed structure of all these compounds, okay? But just pay attention to whatever I'm saying. So it'll go to oxaloacetate, and then oxaloacetate, if you take the reduced form of oxaloacetate, that is called malate, right? So this oxaloacetate can be acted upon by the high energy electrons that you get from NADH, right? So NADH goes to NAD plus, releasing its high energy electrons to oxaloacetate. So oxaloacetate gets reduced to what is called malate, right? So malate and oxaloacetate, they are like redox pairs. You oxidize one, you get the other. You reduce the other, you get the original form back, right? Now, what is the difference? Well, malate can go through the cell membranes, right? It can go through the membranes of the mitochondria and the vacuole and so on, right? So it can be transported from the cytosol to inside the mitochondria, right? So once it becomes malate, it can go to mitochondria and it can go inside vacuoles. So just keep this in mind, because the next process, right, that I'm going to describe, that takes place inside the mitochondria, right? So you can see that, this phosphoenol pyruvate, right, it can get carboxylated to OAA, and then it can get reduced to malate, right, which can then migrate to the mitochondria, right? And this malate in the mitochondria is going to interact with that pyruvate that went inside the mitochondria, right? And that's how the next reaction is going to get going, where there is, oxygen is involved. Okay, so just keep this in mind. Now, any questions so far? Now, no. OA is not in presence of oxygen. It is happening in the cytosol, right? It is carboxylation. Okay, so carbon is being added, right? So this is actually, see, phosphoenol pyruvate is a 3 carbon molecule, right? Oxaloacetate OA is 4 carbon, right? So it is getting more enriched in carbon, right? The oxygen will come into the picture later on. Yeah, go ahead. Yeah. Some of it might go. See, that's why there are controls here. This is an important thing to point out, because all these reactions can take place. Okay, what decides? Okay, whether it is going to go from phosphoenol pyruvate to pyruvate, then to lactate, right? Or whether it is going to go to the mitochondria, or whether it is going to go to the OA. It's about as complicated a control system that you can think of, because there are a lot of controls here. If you are looking at animal cells, okay, see, upstairs, after this, let's see, fructose 6-phosphate, all right, there was an enzyme here that was called phosphofructokinase, right, that I talked about last time, all right. That's the one that gives you fructose 1,6-bisphosphate, right. That enzyme is regulated by the downstream products, right. Like, see, PEP, for example, PEP is going to negatively regulate that. So there is feedback inhibition, right. In the case of plants, PEP negatively regulates phosphofructokinase. So if too much of PEP is being produced, it is going to try to slow this pathway down. Okay. In the case of animals, it's the ATP-ADP ratio, right? Because remember, when do you need to run glycolysis? Exactly, when your energy level is low, all right? So if ATP-ADP ratio is high, that is going to negatively go and negatively regulate phosphofructokinase. Right? So there are lots of controls all over the place. I will discuss the ones I can as we go along, and this is true even in the case of plants. It's not so much the ATP-ADP ratio; it's the ratio between phosphoenol pyruvate (PEP) and the inorganic phosphate ion. Okay. So it's different, right? But again, the underlying philosophy is the same. If too much is being produced, then you need to negatively regulate that. Okay. And again, I mean, it will depend on the needs of OA, because I'm going to show you that in the next step, right, inside the mitochondria, both pyruvate and that OA are going to be needed, right? Now the OA cannot go inside the mitochondria just like that, okay? It has to go through the mitochondrial membrane. For that, it's getting converted to malate, right? Then malate will go inside the mitochondria, get reconverted into OA, right? And that is going to participate in the next reaction that I'm going to talk about, right? All right, any questions so far? So next, I just mentioned, because we talked about 4 different processes, okay? Glycolysis, pentose phosphate pathway, the TCA, so-called TCA cycle, and the oxidative phosphorylation. So I'm going to briefly mention what happens in the pentose phosphate pathway, and then I will show you the TCA cycle that takes place in the mitochondria, right? Just to be, keep continuity with what we were talking about, right, last time. So the second procedure is the pentose phosphate pathway. And this pathway takes glucose 6-phosphate to ribulose 5-phosphate, right? Remember, ribulose is a 5-carbon sugar, right? Glucose is a 6-carbon sugar. So this ribulose 5-phosphate, right? In most organisms, this is the molecule that gives rise to ribose, deoxyribose, and so on, right? So in most organisms, this provides 5-carbon substrates needed for synthesis of ribose, deoxyribose. In plants, however, the ribose and deoxyribose are obtained by a different mechanism that is not fully understood as yet. Okay. So this is very brief. The discussion of the pentose phosphate pathway for us is very brief. We are just saying that this is giving you a source, how starting from the 6-carbon sugar, right, like a hexose, right, you are getting a pentose for synthesizing nucleic acids and so on, right? But I want to next move on to the what is called the TCA cycle, right, or the tricarboxylic acid cycle. So TCA cycle. This is called the tricarboxylic acid cycle, or it is also called the Krebs cycle. And I believe the person who discovered did get a Nobel Prize, right? Now the TCA cycle takes place. So I mentioned that the pyruvate over there may not go to lactate or to alcohol, right? No fermentation. But so if oxygen is available, it is going to move to the mitochondria, right? So this cycle is going to take place in the mitochondria, right? So let's talk a little bit about the mitochondria, right? So this TCA cycle takes place in the mitochondrial matrix, and I will explain in a moment what that is. Right. So if you look at the mitochondria, right, just like the chloroplast, right, it has also got 2 membranes, right? So if you're looking at the mitochondrion, it'll have 2 membranes. One is the outer membrane, right? And then you have an inner membrane which is very, very corrugated, right? So you'll have an inner membrane that looks like this, all right, like lots of folds all over the place, right? So this is the outer mitochondrial membrane, and that corrugated one is the inner mitochondrial membrane. The region inside the inner mitochondrial membrane, that is called the matrix. So this is the matrix, right? And then the space between the two membranes, that is called the inner, that is called the intermembrane space, right? So this is the intermembrane space. Remember, the mitochondrion is the site where most of the cell's energy is generated, right? And I mentioned that most of the cell's energy. Why? Because some of the energy can be generated by glycolysis, which although it's very inefficient, it can produce some energy, right? But most of it is generated in the mitochondrial matrix, right? So how does that happen? Okay, before that, any questions so far? So pyruvate enters the mitochondria and is oxidized via the TCA cycle. And I will explain to you in a moment what are the steps involved, at least the important steps. Now, before that, I just want to point out, I want to go back to glycolysis and point out to you that, suppose you have, energy, like you need to produce glucose, okay? Let's say there are some cells that can only use glucose, all right? And let's say you have the end products of glycolysis, right? But you want to, in other words, if you want to reverse glycolysis, right, can you do that? Well, I mean, some of the steps here are reversible, right? But this pyruvate kinase reaction, right, this reaction here, phosphoenol pyruvate going to pyruvate, that is not reversible, right? So if you could find something, right, to generate the PEP, phosphoenol pyruvate, without having to get it from the pyruvate, you could in theory reverse those steps, right, and produce new glucose, okay? And so wherever you had a kinase, if you're trying to reverse the action, you're going to have a phosphatase, yeah. So in theory, you can reverse those steps, right? However, this step, the pyruvate kinase reaction is not reversible, right? So just keep that in mind, okay? Because we'll visit that later, and that production of new glucose is called gluconeogenesis, right? So one way to do this, okay, one way to do this is if you had OAA, oxaloacetate, like here, right? If I could find, this was PEP carboxylase, right? If I could find something else that just that does just the reverse, you could go from OAA to pyruvate, right? And then move up this chain, right? And ultimately get glucose, right? Produce new glucose. So I'll talk about that, but because I have the figure here, I wanted to just mention this so that, when I start talking about that, you're able to connect everything together. All right, any questions? So now let's discuss the TCA cycle. So pyruvate enters the mitochondria and is oxidized via the TCA cycle, right, or the citric acid cycle. So what are the steps involved? And again, there are many reactions over here. We are not going to discuss all those reactions in detail, but we will point out what is important. Step number 1, right? So pyruvate gets converted to a molecule that is called acetyl coenzyme A, right? We have talked about this before, right? Remember in an earlier chapter when we were looking at a review of organic chemistry, we talked about acetyl coenzyme A, right? So now how many carbons does pyruvate have? I mentioned that earlier, like when you're going through glycolysis, pyruvate has how many carbons? 3 carbons, okay? So just keep that in mind. So 3 carbons. This guy, acetyl coenzyme A, has actually 2 carbons, right? Which means that some carbon has got to be lost, right? And this reaction is catalyzed by an enzyme which is called pyruvate dehydrogenase. So that's the first step. Once it gets inside the mitochondria, pyruvate is converted to acetyl coenzyme A, right? And this procedure is called the oxidative. Carbon is being lost, so it's decarboxylation of pyruvate. Now, since somebody brought up the issue of controls, okay, if the TCA cycle is running, too much of energy is being produced, an indicator of too much of energy is going to negatively regulate this pyruvate dehydrogenase, right? It will negatively regulate that so that the pyruvate is not going to go to acetyl coenzyme A, because that's the procedure. The TCA cycle is the one that produces a lot more energy, right? So instead it might go to glycolysis, or it might even go to OAA, right? So there are controls in there, right? So this is the first step. So pyruvate goes to acetyl coenzyme A. This acetyl coenzyme A is important because, as I'm going to discuss, a little bit later, if you have a fatty acid, see, if you start from glucose, you go down to pyruvate, then pyruvate is going to acetyl coenzyme A. If you have a fatty acid, let's say you don't want to use glucose to produce energy, you're going to get it from a fatty acid. Guess what? The starting point for the fatty acid, right, is acetyl coenzyme A. So you have to somehow from fatty acid, if you get to acetyl coenzyme A, then after that part, the pathway is the same. It doesn't matter whether you started from glucose or you started from a fatty acid or started from a lipid. Okay, and I'll point that out. But I'm just mentioning that these are the things that are coming to complete the big picture, right? So the second step, step 2. So I'm pointing out only the reactions that you just have to know, right? The step 2 is that acetyl coenzyme A that you got from pyruvate. It reacts with OAA, right? Again, OAA is there inside the mitochondria, right? So it reacts with that, and it produces citrate, right? And the enzyme that does that is called citrate synthase. Now, acetyl coenzyme A has how many carbons? 2 carbons, all right? What about oxaloacetate? 4 carbons, all right? So you expect that the citrate is going to have 6 carbons, yeah. Right? The next step of this reaction is that citrate is converted into isocitrate, which is an isomer of citrate, right? So citrate gets converted to isocitrate, and the enzyme that does that is called aconitase. I'm not asking you to memorize all these enzymes, okay? Just for the sake of completeness, I'm writing that down, okay? Then in the next step, isocitrate is converted to what is called alpha ketoglutarate. And the enzyme that does that is called isocitrate dehydrogenase. Notice that each of these steps is catalyzed by an enzyme. That might be negatively regulated by some of the products downstream, and those are known. See, if you're looking at metabolic pathways, a lot of stuff is known because these things have been worked out completely. If you're talking about pathways in cancer or other diseases, we don't know. Okay, we have only partial knowledge. These people have, gone and done complementation tests, all right, put a mutation and all that, and these have been figured out completely, right? What controls what, people know that, right? But it's a multivariable control problem, right? All right, so this gives you alpha ketoglutarate. Then you have a whole bunch of steps, right? You have a whole bunch of steps, and you ultimately get OAA. Remember, this is a cycle, right? So this is a cycle. That means whatever you use up in the beginning, right? So you're processing something, but whatever you use up in the beginning, you're going to have to regenerate that, right? So here I got from pyruvate, I got acetyl coenzyme A. That's the one I want to oxidize to carbon dioxide and water, right? And capture as much energy as possible. To do that, I needed OAA. So I have to ultimately generate OAA, right? So ultimately get OAA. So to keep the cycle going, for continuing the cycle, right? And there are different reactions, all right? There is fumarate, succinate, and so on. But the important thing to note is that this reaction is going to produce carbon dioxide, right? Because you're trying to oxidize carbon, right? Then it is also going to produce NADH, right? Which has high energy electrons, all right? Then it also produces another molecule that we have not discussed, but it's similar to NADH. It's called FADH2, right? It has flavin. I mean, the detailed composition is different. Function is similar, right? And it also produces ATP. So ATP being produced along the way. right? So this is a description of the TCA cycle that takes place inside mitochondria, right? And you basically have to keep on. So every cycle, right? This acetyl coenzyme A is getting used up, right? You're getting carbon dioxide, right? And the OA is being generated, so the cycle can continue. Okay. Now the OA that is in there, right? Along the way, right? It goes through malate. Remember, malate is the reduced form of OA, right? What property does it have? If it is malate, it can get out of the mitochondria, go to the cytosol. So if more OA is needed in the cytosol, what will happen is that the malate will leak out of the mitochondria, go to the cytosol, and get reconverted into OA, right? If that's needed. Okay. So there are lots of controls that are taking place. Right? So this completes our discussion of the TCA cycle, right? And now the thing that remains is we have to see how oxidative phosphorylation takes place. Because now, remember, you have these high energy electron carriers, right? NADH, FADH2, and so on. Now you want to use that to synthesize ATP, because ATP is the ultimate storage of energy in the cell, all right? So some ATP you are getting from the cycle only, all right? You got some from glycolysis, some from the TCA cycle, and now you need what is called oxidative phosphorylation, right? Where basically these high energy electron carriers, all right, they are going to move down a chain, right? They're going to move from unit 1 to unit 2 every time they're losing some energy, and ultimately they are high energy electrons that oxygen is a strong electron acceptor, right? So ultimately they will pass off the electrons to oxygen, forming water, right? And while this is going on, there is proton pumping, right? So that produces basically a proton gradient, right? That can be used then to produce ATP, right? So let me explain again in a little bit more detail. This is similar to what happens in the chloroplast, right? Remember, we had those electrons going down. What was the big difference? The big difference is that in the case of the chloroplast, those carriers were NADPH, right? Here it's NADH, right? Because these are catabolic reactions. These are not anabolic reactions, right? So if you look at the membrane of the inner membrane of the mitochondria. So this is the inner mitochondrial membrane. And so here you have different complexes, all right? So this is complex 1. And again, you can look at the book for the detailed composition of these complexes. That's not important for us. There's complex 2. These are respiratory complexes, then complex 3 and complex 4. So these NADH, right, with the high energy electrons, they will come in here, right? So NADH, right, will come in here, and ultimately you're going to have NAD plus being produced, right? It's going to give up its high energy electrons to that complex 1, right? The complex 1 will give it to complex 2 and so on. Now these guys, all right, while this electron transfer is taking place, complexes 1, 3 and 4, they actually pump. So this side at the bottom, that is actually the matrix. So this is at the bottom is the matrix, mitochondrial matrix, all right? And at the top, it's the intermembrane space between the inner and the outer mitochondrial membrane. So this place is the intermembrane space, all right? So complex 1, 3, and 4, they will pump hydrogen ions or protons from the matrix. So 1, 3, and 4. So again, this is also pumping hydrogen ions from the matrix into the intermembrane space. Okay. So and finally, complex 4 is where oxygen is going to accept the electrons and form water. Okay. So complex 4 is where so this is where electrons passed off to oxygen forming water. To oxygen forming water, because you know that the products of respiration are carbon dioxide and water, right? And again, this reaction, this TCA cycle and all of these steps, they have to be carried out incrementally, because at each stage, whatever energy is being produced, all right, the cell is going to try to capture as much of that as possible, right, in the form of ATP. Otherwise, it's going to be released to the environment as heat. So the proton pumping, right, across this mitochondrial membrane, that is going to produce a proton gradient, right? So if protons flow down that gradient, right, you can couple that to the production of ATP, right? So that's how the energy is produced. So let me write that out. So proton pumping across inner mitochondrial membrane and ATP generation. So this produces the proton motive force, right? And when protons flow down that gradient, right, you have this kind of machinery that is going to produce ATP. And ATP generation via and what is the name of the enzyme, right? Or the molecular machine that is going to produce ATP? ATP synthase, right? Synthase or synthetase, right? And the reaction that you have here is NADH, right, plus hydrogen ions plus half oxygen, right, is going to give you NAD plus, right? That means that is the oxidized form of NADH, right? And then you have water. So oxygen is the ultimate electron acceptor, right? But in the process, you have managed to produce that proton gradient across the membrane, and then when protons flow down that gradient, you're able to generate energy, right? Now, all right, any questions so far? So as I said before, right, glycolysis produces very small amount of energy, right? So 4 molecules of ATP per molecule of sucrose. Now, if you look at animals, we use glucose, right, for producing the energy. So the number is half because you have a glucose and a fructose will give you 1 molecule of sucrose, right? And if you're looking at aerobic respiration, the number is significantly higher. And you can do the calculation because if you go through each of those reactions, how many ATPs are used up, how many are being produced, how many NADH, when you're going through the electron transport system, you can do the calculation, right? It's not difficult to do, but it's just that I don't have time here to work that calculation out, right? It's there in the book and you can go through the steps and make sure that those numbers are correct. So aerobic respiration produces 60 molecules of ATP per molecule of sucrose. And so if you're looking at animals, that number will be halved, okay? It will be about 30. But you can see it's significantly more than what you would get from glycolysis, right? And I already talked about the Pasteur effect and the Warburg effect. The Pasteur effect is in the absence of oxygen, the plants or even animals, cells will exclusively run glycolysis, right? But if you give it oxygen, then it is going to run the TCA cycle and produce a lot more energy, right? In cancer cells, you have what is called the Warburg effect that was discovered by Otto Warburg in the 1920s, right? And he got a Nobel Prize, I think, for that discovery. And that was that cancer cells, even in the presence of lots of oxygen, they run glycolysis, right? And one reason could be because the intermediates of glycolysis, it is in terms of energy efficiency, it is low, right? But the intermediates of glycolysis, they provide valuable substrates for producing new cells. All right, any questions so far? So this is like carbohydrate metabolism or sugar metabolism we discussed. I have one more thing to discuss, that is how you can produce new sugar, right? This you're talking about breaking down sugar, right? But let's say you you have certain cells in the body that can use only sugar, right? Like in the case of animals, the red blood cells can use only sugar. So if you need sugar and sugar is not there, your blood sugar is low, right? There are controls that will make sure that other molecules are broken down to produce the sugar that you need, right? So next I want to talk a little bit about lipid metabolism and also lipid synthesis. So lipid metabolism. Now lipids are compact, high energy storage, right? Because if you have too much of glucose in your blood, initially, the excess glucose will get stored in your body as glycogen, but that has too much of water, right? So it's not compact storage, and then the production of fats will start, right? That's why we gain weight. I mean, like if we eat too much, right? If you're eating more than what you need to expend, right? Then in the steady state you'll start gaining weight, right? So lipids are compact, high energy storage, right? So and lipids are made up of what kind of molecule? What? Phospholipids is one of them, yeah. So lipids typically in cells, they are stored as fatty acids in cells. What are they stored as? Triglycerides, yeah. So lipids, typically the body fat is triglycerides. So if you want to get energy from these so triglyceride means a glycerol backbone and 3 fatty acids, all right? So first step is to cut out the fatty acids from the from the triglyceride, right? And then try to oxidize them to get the energy, right? So triglyceride, so typically the triglyceride will be broken down by an enzyme that is called lipase. Again, ase means to break down, okay? Lipase, lipid breakdown. So this is going to give you a glycerol backbone, right? Plus it's going to give you 3 fatty acids, right? And now you're going to oxidize these fatty acids to get carbon dioxide and water and of course a lot of energy, right? So how does the oxidation of fatty acids proceed? Well, the first step is what is called beta oxidation of fatty acids. They undergo beta oxidation and that gives you acetyl coenzyme A, and you do get some other NADH and FADH2 also. So if you're calculating the detailed energetics for these reactions, you have to keep track of all these guys, right? To find out how many molecules of ATP are produced. So the beta oxidation of fatty acids gives you acetyl coenzyme A, right? Now this acetyl coenzyme A now can enter the TCA cycle, right? It doesn't matter where it came from. Did it come originally from glucose or fructose, or did it come from the fatty acid? It can go into the mitochondria, right? And go through the TCA cycle and be processed exactly the same way that a molecule of sucrose would be processed, right? Would be ultimately processed. Do you see that? And in the case of plants, this reaction, right, this processing, this beta oxidation, it is carried out inside an organelle that is called a glyoxysome. So carried out inside a glyoxysome. In the case of animals, it's going to be carried out inside mitochondria. So this is how lipids are broken down to produce energy, right? So in one shot, I have covered both lipids and carbohydrates because I'm showing you that there's a common meeting point, right? After that, it's exactly the same, right? It doesn't matter whether you started out with a carbohydrate or you started out with a lipid, okay? Any questions? Again, I'm trying to focus on the big picture, right? You're not going to learn the details of all the reactions. You don't need to. If you're going to work on, let's say, the control of, lipid metabolism or something, then you will get down into the trenches and dig through the books, all right, and the papers, and figure out what all the controls are, and you could do pretty interesting modeling, right? Some of you are looking for topics. This is one of them, right? You can look at that. What have people done so far? Okay, can you improve things, right? Uh, so next I want to talk about how are fatty acids synthesized, right? Synthesis of fatty acids. And there are a lot of controls at play, okay? Because the cell needs to decide, both in animals and plants, okay, should it be synthesizing fatty acids and forming triglycerides? Or should it be breaking down triglycerides and doing beta oxidation of fatty acids to get energy, right? So obviously the energy needs of the moment will play a critical role, right? Like they tell you, if you work out more, you're going to lose weight, right? You're going to break down fats and all that. Because you need more energy, you just don't have it, you have to break down stuff. On the other hand, if you don't exercise, you just keep on eating. You're in a feasting state, right? Then if everything else is working fine, you have excess energy, you need to store it, you're going to put on fat, right? Or put on weight. So let's talk about the synthesis of fatty acids. Again, that starts with acetyl coenzyme A. Acetyl coenzyme A. You add carbon dioxide to that. Again, how many carbons does acetyl coenzyme A have? 2 carbons, okay? Alright? So 2 carbons plus carbon dioxide, right? I mean 2 main carbons, okay? There might be other carbons in the other portion, okay? Because it has got a long tail, okay? But 2 main carbons plus carbon dioxide. So this is a carboxylation reaction, right? So you will get a molecule with 3 carbons, alright? And so this reaction actually uses ATP, and the enzyme that does this is called acetyl coenzyme A carboxylase. This gives you something that is called malonyl coenzyme A, right? and this actually has 3 carbons. All right, this has 3 carbons. Now, so the first step in fatty acid synthesis is the formation of malonyl coenzyme A, and this is the rate limiting step, right? Like if you want to stop the synthesis of fatty acids, the negative regulators will actually negatively regulate acetyl coenzyme A carboxylase. All right, it will stop the production of malonyl coenzyme A. All right, in the next step, what happens is that acetyl coenzyme A is going to react with malonyl coenzyme A that you have just produced, right? And then there will be other stuff too that go in, like NADPH plus H plus and so on, right? And it produces a fatty acid 1. Let's call this fatty acid 1 plus carbon dioxide and so on. Okay, let's not worry about the other things. The important thing to note is that this malonyl coenzyme A, right? Every time you go through this second reaction, so the first reaction, you need the malonyl coenzyme A to get this reaction started, right? Now in the next step, acetyl coenzyme A, now acetyl coenzyme A has how many carbons? 2 carbons, all right? All right? So what it's going to do is it's going to react with malonyl coenzyme A, and it is going to produce a fatty acid that will have 2 more carbons than what it had before. Okay, so if you had 0 carbons, now it's plus 2, right? Then in the next step, again, this reaction will continue, right? So so you take this fatty acid number 1, so fatty acid 1, and then you make that react with malonyl coenzyme A plus NADPH plus hydrogen ion. It'll give you the second fatty acid, right? Fatty acid 2 plus carbon dioxide, right? The important thing to note here, not the detailed reactions. Look, malonyl coenzyme has how many carbons? 3 carbons, all right? So every time you're using this malonyl coenzyme A, right, taking a fatty acid, make it interact with malonyl coenzyme A, you are adding 2 more carbons to the original fatty acid, and the third carbon is being expelled as carbon dioxide. Okay, so you can do this, and usually it goes up to 16 or 18, right? Until you have 16 or 18 carbons, right? But this is the step-by-step synthesis by which a fatty acid is produced, right? And you have enzymes. There's an enzyme that is called fatty acid synthase that will make this reaction go forward, right? So, but the starting point is malonyl coenzyme A. Malonyl coenzyme A will react with acetyl coenzyme A, give you some fatty acid plus carbon dioxide. Take that fatty acid, again make it interact with malonyl coenzyme A, same kind of reactions, all right? Then you will get a fatty acid that has got 2 more carbons, all right? And then carbon dioxide. Right? That's how fatty acid is synthesized. Now what decides whether synthesis will take place or breakdown? That will be the energetics, the needs of the moment, right? Okay. So this procedure usually stops. So stops when carbon length becomes 16 or 18. And it can stop even before that, but I mean 16 or 18 is probably the maximum number that you usually see in nature, right? Now the lipids, right, they make up the cell membranes, right? Now when you're looking at these fatty acids, right, they can be either saturated or they can be unsaturated, right? So saturated versus unsaturated fatty acids. Okay, what's the difference? Yeah, so unsaturated fatty acids have double bonds, okay? Now if you have like some oil that has got a lot of saturated fatty acid in it, right? Then at room temperature it will be solid, right? I mean you can think of, fatty acids that are saturated, right? I don't know what it's called here. For example, in India there's something that they call dalda and all that, and that's frozen. That is really saturated, heavily hydrogenated fatty acid, right? So that's frozen. That's bad for you. I mean, health-wise, that's supposed to be bad for you. The unsaturated or polyunsaturated fatty acid, that's good for you, okay? Because over there, because of the double bond, the packing is not that tight, right? So there is more flexibility in the membrane. So naturally, if you have plants, okay, they have membranes. Let's say they are made of fatty acid. If the temperature falls, all right, It's going to freeze. So plants have mechanisms by which maybe they will change the composition of the fatty acid in the membrane from saturated to unsaturated, right? So that they become chilling resistant, right? Because some plants they die when you have this winter freeze like we had a few days back, right? Some others don't, right? And this ability has something to do with that, right? So saturated versus unsaturated fatty acids and their role in chilling response, right? Or chilling resistance. And again, it's not just saturated versus unsaturated. Even the particular unsaturated fatty acid that you have, that can also be a determinant about whether the plant is going to freeze to death or not. Okay, because remember, inside the plant you have all these tubes, right? These vascular bundles and things like that. Okay, and there is a lot of membrane transport that has to go on, right? All the time. So if something freezes, then that's a problem because then it cannot get the molecules that it needs. So ions and things like that, the water transport, all that is usually disrupted, right? All right, any questions? So I have covered glycolysis, I've covered TCA cycle, beta oxidation, right? Then I also covered lipid synthesis. Again, it's the big picture, it's the overview. Not much memorization, maybe a little bit, but not much, right? Because each one of these has got many steps in there. There are controls in there, right? What is controlling what? There are things that are linked together, right? And it's a multivariate thing. So it's not that easy to just go and study the process in isolation. The last thing that I want to cover in this chapter is what is called gluconeogenesis or reversal of glycolysis. All right, before that, any questions so far? So, as I said before, when you're looking at glycolysis, that pyruvate kinase reaction is not reversible, right? So the other steps you can reverse, right, by using phosphatases instead of kinases, right? So one way so let's say you have too much of these products of glycolysis, or you just don't have, let's say raw glucose, all right? Your cells need glucose. As I said, the red blood cells in the case of animals, they use only glucose, right? They don't use fatty acids. So if you don't have any glucose, if your red blood cell, your glucose level goes down, then you have to break down proteins and fats and try to uh, produce glucose that you need, right? So the plants do that. Like for example, in the case of seeds, right? The nutrition might be stored in the form of lipids, but when the embryo develops, it needs sucrose, right? Sugars in order to grow, right? So there has to be a process for converting, let's say, lipids into sugar, right? And that is there, and that is called gluconeogenesis. Same thing, even for proteins, also it is there. Like if you start working out too much, right? You've first exhausted all your glycogen, right? Your fats, then you'll start losing muscle mass, right? Because your proteins are going to break down, and proteins basically, they will have the backbone like the carbohydrate, but then there's an amino group somewhere. So the amino group will get removed, and then this is going to produce glucose that can then be used by the red blood cells and so on. Okay. So let's talk about gluconeogenesis. All right. So the starting point, one way to bypass that pyruvate kinase reaction, right, is to basically find out a mechanism that can produce PEP, phosphoenol pyruvate, the step above the pyruvate kinase reaction, right? And you can do that because just like PEP could go to OAA, right? Like I showed you, it would go to OAA, then to malate, and then move to the mitochondrion. Same way, malate could leak out of the mitochondrion, come out, get converted to OAA, right? And you'd have to have an enzyme that will take OAA to PEP and then drive glycolysis in the reverse direction, right? So in gluconeogenesis, and again, starting from a protein also you can do that because if you take the amino group off from some amino acid, you can get OAA and then the glycolysis reaction can run in reverse, all right? So if you have OAA, right, and it is treated with an enzyme that is called PEP carboxykinase, then you get PEP phosphoenol pyruvate plus carbon dioxide, right? You have to get a carbon dioxide because OAA has got how many carbons? 4 carbons, all right? So this has got 4 carbons, and then PEP has got 3 carbons, all right? So you will have to have carbon dioxide removed. So this is like decarboxylation of oxaloacetic acid, right? So this step, right, this reaction, and again, you will need that kinase, this enzyme. So the enzymes will decide whether the reaction is going to go from PEP to OAA or from OAA to PEP, right? And the needs of the moment will dictate which enzyme is going to be produced. Remember, we talked about the lactose operon, the tryptophan operon, and so on, okay? Whatever, depends on the needs of the cell, it is going to produce more or less of one enzyme or the other, okay? So this step actually bypasses the irreversibility of the pyruvate kinase reaction. Alright, so if you have OA readily available, you can run glycolysis in reverse, and that is called gluconeogenesis. Right, and the OA could come, it could come from even from proteins or from amino acids. It could also come from lipids. Okay, amino acids. How? If you put an amino group on OA, you get some particular amino acid, which I'm going to talk about later, a little bit later. If you remove amino acid from, if you remove the amino group from that amino acid, you will get OA. Right. You can do the same thing for lipids, but for lipids, remember when you do beta oxidation of lipids, you get acetyl coenzyme A, right? Acetyl coenzyme, you cannot produce glucose from acetyl coenzyme A. You have to run that acetyl coenzyme A through the TCA cycle up to the stage where malate, which is the reduced form of OA, is produced. That malate can move outside the membrane because it can move through the lipid layer by layer, come into the cytosol and get oxidized to OA, right? Which can then be used. So in other words, the source of OA does not have to come from the glycolysis. It could come from protein breakdown, it could come from lipid breakdown and so on, right? So let's talk a little bit about the lipids to glucose. Okay, how do you produce glucose from lipids? And this happens in seeds because the stored nutrition is in the form of lipids, right? But the seeds need glucose to germinate, right? So how does that conversion take place? So lipids to glucose, and this happens in plant seeds. And the first step is that if it's a lipid, it's a triglyceride. So lipase, some lipase, right, is going to release those 3 fatty acids, right? So releases fatty acids from triglycerides, right? Then after that, you have the beta oxidation of fatty acids. So the beta oxidation of fatty acids will give you what? If you do beta oxidation, if you break down fatty acids, okay, using this procedure called beta oxidation, you get acetyl coenzyme A, right? I mean, so acetyl coenzyme A, and then acetyl coenzyme, and usually these reactions, all right, in the case of plants, they occur inside glyoxysomes, all right. In the case of animals, they occur inside mitochondria, right? So the lipid has produced acetyl coenzyme A, okay. Then it can run through the TCA cycle, right? It can run through the TCA cycle, and then along the way, right, you're going to produce malate somewhere, okay. That can come out and then get oxidized back to OAA out in the cytosol, all right. So the reactions of the TCA cycle will take place one after the other. And in the case of plants, this will be running inside the glyoxysome. So acetyl coenzyme A plus OAA will give you citrate, right? Then citrate will go to isocitrate. And I gave you the name of the enzyme and so on. There are a number of steps here. Then you will have succinate is produced. Fumarate is produced. These are the different molecules that are produced one after the other, a number of steps. Then somewhere there is malate that is produced, right? And that malate can go to OAA. The malate can leak out of the mitochondrial matrix, mitochondrial membrane, go to the cytosol, right? Over there, it can get oxidize to OAA, right? And now this can enter gluconeogenesis, run glycolysis in reverse, all right? So it can produce OAA in cytosol. So this malate can leak out of mitochondria. Actually, here it will be glyoxysome, so not mitochondria. So here, I mean, to summarize, for gluconeogenesis from lipids, you will be running the same steps as in the TCA cycle. So this is carried out in mitochondria in mammals, but in glyoxysomes for plants. See, if you are if you need to produce more glucose, right, from lipids, right, you're not going to be trying to run the TCA cycle, right, because you want the stuff to go back to glucose. So in the TCA cycle, you'll go to the step where malate is produced, then that will leak out, right, because that is more important. Let's say you're in the case of the human body, your red blood cells need energy. They are only going to survive on glucose. You might have tons of lipids lying elsewhere, right, but you need to quickly convert that into glucose, right, just to survive, because if your red blood cells give up, then your other cells will not get oxygen, you'll die, right? So depending on the needs of the moment, right, the reaction can go either way, right? In one case, like even for lipids, you're synthesizing in one case, right? Doing lipid synthesis using malonyl coenzyme A and so on. And in another case, you're trying to break down lipids to get energy, right? So when you don't need too much energy, you store it, right? Once you need energy, you start breaking down your stores, so that you can survive during that lean period. All right, any questions? So now, if there are no questions, so I have talked about respiration and lipid metabolism in this chapter, right? I've tried to give you the overview. Most of the steps are pretty similar in plants and animals. Okay, even if you're looking at animals, you have glycolysis, you have, the TCA cycle and so on, right? Then somebody mentioned last time, like if you're exercising, right? Too much, you're exercising too much and enough oxygen is not getting to your cells. Then you experience burning sensation. That's because in that case you have lactate that is being produced, right? The pyruvate is not getting enough oxygen, it is not going to the TCA cycle, lactate is being produced, right? And lactate is acidic, right? So that's why you experience the burning sensation. But there are other ways. The lactate can then go to the liver, right? And over there it can be used to produce glucose and so on, right? So all these things are linked together, right? And I don't have time to cover all of this in this particular class, but I can at least show you what the main connections are, so that depending on what your interests are, if you want to focus on animal biology, right? You can look at that, okay? But the steps are the same. Keep in mind just a few things: ATP, NAD, NADPH, and so on. Okay, whether you look at plants or in animals, things happen the same way. All right, any questions? Otherwise, we move on to the next chapter, which is chapter 13, which is about assimilation of inorganic nutrients. So basically, we are going to talk about assimilation of nitrogen and sulfur. So plants need to absorb both nitrogen and sulfur. These are both essential elements. Okay, nitrogen is needed in amino acids, then it's needed in nucleotides, right, to make nucleotides. And sulfur is actually needed for making cysteine, methionine, and so on, some of the amino acids. So sulfur will be in cysteine, methionine, and nitrogen is needed for so many things like amino acids, nucleotides, etc. Even this NADH and NADPH, they also contain nitrogen. Now, fixing nitrogen is a very, very high energy consuming reaction, right? So taking nitrogen, so the bond for nitrogen is extremely stable. Okay, if you take atmospheric nitrogen and you try to fix it, it requires a lot of energy. Like some nitrogen gets fixed when you have strikes of lightning during thunderstorms. Okay, so or industrial processes fix some amount of nitrogen. So nitrogen fixation is a very, very energy consuming process. So nitrogen fixation is very energy consuming. And if you look at the total amount of nitrogen that is fixed, atmospheric nitrogen, about, let's see, 8% is fixed by lightning, right? So nitrogen gets converted like into ammonia, right? Then about 2% is fixed by photochemical reactions. And the remaining 90% is fixed by bacteria, right? Bacteria in the soil, right? So 90% by bacteria. Now, to give you an idea, see, I said that this business of producing, atmospheric nitrogen, converting it into ammonia, right? Then to nitrates, right? Then maybe ammonium ions, amino acids. These are all very, very high energy absorbing reactions. They require lots of energy, right? So if you run the reaction in reverse, what is going to happen? Suppose you have nitrate, right? Ammonium nitrate, and you're going to get nitrogen from it. What is going to happen? It's going to release tons of energy. Okay, that's why it's not surprising that all these explosives and all that, they have nitrogen in them, right? Because just the reverse reaction takes place. Because if you're putting in so much energy into generating that molecule, if you reverse the procedure, it is going to release tons of energy, right? Now, so if you have nitrogen that gets fixed into ammonia, right, then ammonia can react with water to produce the ammonium ion plus the hydroxyl ion, right? Now, unassimilated ammonium, right, or nitrate may be dangerous. So unassimilated ammonium ion or nitrate may be dangerous. In fact, I'm going to show you now that if you have ammonium ion, right, what it can do is it can dissipate the membrane potential. See, like there are certain things in the cell for which you do need that proton gradient, right? That pH difference across the membrane. If you're going to do ATP synthesis, okay, you need that pH difference. If you're going to transport some molecule using a symporter, you need that pH difference, right? So for various essential cellular activities, you need to maintain that pH difference across the cell, all right? But if you have an ammonium ion, right, injected into the cell, it can flow freely across membranes and it is going to mess everything up, all right? So let me show you using a picture how that is going to happen. So suppose this is a membrane. On the left-hand side, you have a high pH, right? And on the right-hand side, you have a low pH, right? And this has been achieved by proton pumping. Like we have in photosynthesis, then even in respiration, we have proton pumping reactions, okay? So high pH means, what about the hydrogen ion concentration on the left-hand side? If the pH is high, that means? So low hydrogen ion concentration, right? And on the right-hand side, it's high hydrogen ion concentration, right? Now, ammonium ion is going to diffuse freely from one side to the other. If you look at the left-hand side, what it's going to do is ammonium ion will react with the hydroxyl ions on the left-hand side, and it will give you the gas ammonia, and a molecule of water is removed, right? So look, on the left-hand side, you have used up a hydroxyl ion, okay? So what is going to happen to the pH? Is it going to go up or is it going to go down? Go down. You've made it more acidic. So this is going to lower pH, right? Right. On the right-hand side, so this ammonia gas, it's formed the ammonia gas. The ammonia gas can now diffuse across the membrane, right? And it reacts with the hydrogen ion, the proton over here, because the right-hand side has high hydrogen ion concentration, and it produces the ammonium ion, right? So what has happened to the pH on the right-hand side? Has it gone down or gone up? Up, right? So can you now see that the presence of the ammonium ion is going to mess things up, because On the left hand side the pH was high, right hand side it was low, right? So this will bring it to a situation where there is no pH difference between the two. So all those essential processes are going to stop, right? The cell will die, right? So this makes the point that unassimilated ammonium or nitrate ion can be dangerous, and that's probably one of the reasons why animals have a strong dislike, right, for the smell of ammonium ion, right? Now plants can store high levels of nitrate and translocate it from tissue to tissue. Now for animals and humans, that's a problem, all right? If you have high levels of nitrate, or if you take food containing too much nitrate, you're asking for trouble, okay? The first thing is that for animals, right, the nitrate can get converted to nitrite, right? So for animals, you can have nitrate will go to nitrite, right, in the liver. And this nitrite, it will bind hemoglobin. So nitrite it binds hemoglobin, right? So what is the normal function of hemoglobin? Huh? It's supposed to bind what? Oxygen. Yeah, if it goes and preferentially binds these nitrites, okay, then it's not going to be able to do its job of binding oxygen, right? And it can lead to this disease that is called, let's see, METHE, methemoglobinemia, right? Right, because your hemoglobin is not active, it's not being able to do its work, right? That's one possibility. In addition to that, it can also cause cancer because nitrate, right, can get converted to what are in animals. It can get converted to nitrosamines, right, which are potent carcinogens. Or it can also get converted to nitrate, right? Nitrate, I'm sorry, not nitrate, nitric oxide, which is a gas which causes your blood vessels to dilate, right? So if they give you nitric oxide, right, your blood vessels will dilate, right? So there are situations when that is necessary, but if it just happens just like that, that's not good, right? So nitric oxide, it's a vasodilator. That means that's something that makes your blood vessels dilate, right? Those are called vasodilators. So how is nitrate assimilated, right? In the case of plants, okay, how is nitrate assimilated? So nitrate assimilation, so nitrate is first converted to nitrite. So nitrate is like NO3 minus, right? There are some other things that that go in, but the net result is that you get nitrite, NO2 minus, right? So this is nitrite, and the enzyme that does that is called nitrate reductase. Is nitrate reductase. See, ultimately, you have to get the nitrate, the nitrogen, you have to get it into an amino acid form. So it is going to get reduced from nitrate to nitrite. Then from nitrite, it is going to get reduced to ammonium, right? And then even in the case of ammonium, it cannot stay there because it's going to mess up the cell membranes. So it has to get converted from there to amino acid, right? So nitrate, from nitrate you'll go to nitrite. Then from nitrite, that is NO2 minus, the enzyme in this case will be called nitrite reductase. You will get the ammonium ion, right? And then you have to assimilate that ammonium ion, okay? Why? Because even the ammonium ion is going to mess up those membrane potentials, right? In the case of plants and animals. So you have to convert it into amino acid. So ammonium ion needs to go into amino acid, right? And I give you several examples of that, okay, how it's going to happen. For example, if you have the amino acid that is called glutamate or glutamic acid, if you add ammonium ion to it, right, and you add ATP, you get another amino acid which is called glutamine plus ADP plus inorganic phosphate, right? You consume ATP, but from glutamate you can get glutamine, right? So glutamate is already an amino acid. It has got amino group, carboxyl group. Then you add some amino group at a particular location, you get glutamine, right? That's another amino acid, right? And the enzyme that gets this reaction going is called glutamine synthase. Then there is another amino acid, and these are important because the way nitrogen is absorbed, both in case of plants and animals, is it'll usually go through these guys, through glutamine, all right? So it'll start with glutamate, so once it gets into glutamine, now that nitrogen can be transferred to other, precursors of amino acids to give you those other 20 amino acids. So usually it's glutamate and aspartate, these are the main entry points, right, for nitrogen. So aspartate, and even for mammals, right? Like they have a urea cycle and all that, how nitrogen is removed and all that stuff. But this glutamate and aspartate play an important role over there. So even aspartate plus an amino group gives you so aspartate is also an amino acid, it's aspartic acid. If you add an amino group to that at a particular location, again, we are not worrying about the details, right? But if you add it to that, then you get this another amino acid that is called asparagine. Now I want to talk to you about the precursors for some of these amino acids, all right? So one of them we have seen before, it's called alpha ketoglutarate, right? I talked about that in the TCA cycle, right? Alpha ketoglutarate, again, you don't need to know the formula and all that, but at a particular location, if you add an amino group, right, to alpha ketoglutarate, then you get asparagine. I'm sorry, not asparagine, you get glutamate, right? Sorry, my mistake. So you get glutamate. If you take OAA, oxaloacetate, right? If you add an amino group, you get aspartate. So see, to get glutamate, all right? If you start with alpha ketoglutarate, add an amino group, right? Then you will get glutamate. If you start with OAA, right, which is one of the things that we saw in glycolytic and TCA cycle pathways, you will get aspartate, right? And actually, even if you take pyruvate, you add an amino group to it, you will get something else that is called alanine, right? So I didn't have that in my notes, but let me just mention that to you. So pyruvate, if you add amino group, you put an amino group on it, you will get alanine, and so on. Okay. So, if you need the amino acid alanine, all right, guess what? The pyruvate is not going to go through that lactate and all that stuff. The pyruvate is going to get an amino group added and it's going to produce alanine. Similarly, breakdown of alanine could produce pyruvate, right? That can then be used to generate energy through the glycolytic pathway, right? So all those things are intertwined, all right? Now I want to point out, so these two things, all right, aspartate and glutamate, they are very important. They are like the entry points for nitrogen, right? Whether it is animal cells or plant cells, or roughly speaking, I don't know about all the, myriads of reactions, but whatever I have seen, they are the entry points, all right? So now if you have the amino group on aspartate or glutamate and you want to transfer it somewhere else, there are reactions that are called transamination reactions, all right? Where, say for example, if you started with, let's say you started with glutamine, all right? You transfer the amino group to something else, you get the corresponding amino acid, and then glutamine will become glutamate, right? Because glutamate plus amino group is glutamate, right? Glutamate plus amino group is glutamine, right? So let me talk a little bit about these transamination reactions. Yeah, you don't have to remember anything much. Only thing is that just keep in mind which ones are the pairs. Like alpha ketoglutarate and glutamate, they form a pair, right? You can go from one to the other by adding an amino group. OA and then aspartate, they form a pair, right? Just keep that in mind, right? So these two are important, right? These two and even this glutamine and asparagine, these are important. But remember, there are 20 different amino acids, but I'm just talking about 4 of them. Okay, because these show up again and again, right? They are like the entry points. So say for example, you have in a transamination reaction means amino group will move from one amino acid to another, right? So for example, if you have a reaction, let's say glutamine, right, plus aspartate, right? And glutamine is going to transfer, right, its amino group to aspartate, right? So what are the products going to be? This is a transamination reaction that is catalyzed by the enzyme that is, anyway, I'll give you the name of the enzyme in a moment. But what are the products here going to be based on the information that I've given you? If glutamine gets rid of its amino group, what amino acid are you going to get? Glutamate, okay, good. And if aspartate, right, gets that amino group from glutamine, okay, what amino acid are you going to get here? Huh? Go ahead. No. Aspartate is getting an amino group, right? OA you will get if you take the amino group away, right? If aspartate gets an amino group, what amino acid are you going to get? Asparagine, all right? Right? So this is an example of a transamination reaction, right? Amino group transferred from one amino acid to the other. And this, the enzyme that makes this reaction happen is called asparagine synthase. And it uses ATP. Remember, to make reactions happen, you need enzymes. You also will need energy. Okay, so this reaction needs energy in the form of ATP, right? Let me give you another example, right? So if you have glutamine, right? Let's say glutamine, and then you have alpha ketoglutarate. So what are you going to get here? If you have glutamine and then alpha ketoglutarate, glutamine is going to transfer its amino group to alpha ketoglutarate. So what are you going to get? First of all, when glutamine gets rid of its own amino group, what molecule do you get? What amino acid do you get? Glutamate, all right? Yeah, good. And alpha ketoglutarate gets that amino group, and again it will become one more glutarate, all right? Glutamate. So you're going to get 2 glutarate, right? So 2 molecules. See that it's not that difficult, right? I mean, it's quite straightforward. But again, I mean, there are lots of these that are taking place, and keeping track of that, which reaction will take place, we don't know, right? But this stuff is not that difficult, right? Now, yeah. So now, how does the biological nitrogen fixation take place, right? That is carried out by bacteria in the soil. So there is a symbiotic relationship between plants and the bacteria in the soil, right? So these will, and there is signaling that goes on. The bacteria in the soil will send out some signals to the plant will respond, right? So the signaling system, and then this signaling causes the roots of the plant to develop some nodules, right? Because this nitrogen fixation reaction, it has to be carried out in an anaerobic environment, right? Why? Because this is a very high energy reaction. A lot of high energy electrons are exchanged. Oxygen is a very powerful electron acceptor, so it's going to interfere with those reactions. So nitrogen fixation means nitrogen in the air is going to be converted into ammonia, right? Nitrogen and hydrogen, they'll be put together, giving you ammonia. This needs to be carried out in an anaerobic environment. So nitrogen fixation must take place in an oxygen-free environment. So the interaction between these bacteria that fix nitrogen, right, and the roots of the plant produces nodules on the roots, right? And it has some kind of a sac where there is no oxygen, and this nitrogen fixation can take place. Right? These bacteria get into the roots, and then they fix nitrogen. They produce ammonia, which is then converted into nitrates and then assimilated by the plant. Right? And then the last thing that I want to talk about is sulfur assimilation and phosphate assimilation. Sorry. So sulfur gets in the soil. It's present in the form of sulfate, right? And so sulfate ion, and phosphorus is in the form of the phosphate ion, right? Sulfate, and then phosphorus will be phosphate ion. Now, how are these absorbed? Both of them, it's similar. Okay, these are negatively charged ions, all right? So remember, in the case of the roots of the plant, the exterior has got a higher pH, all right? Because remember, in the apoplast, inside the cells, protons are being pumped out, right? So it will make the environment acidic. So then the sulfate is a negative ion, okay? So because of that pH difference, all right, the protons have a tendency to flow from outside to inside, and these guys can hitch a ride, right, when that is flowing from outside to inside. So both of them, basically the transport involves hydrogen ion symporters. Remember, I talked about things going in the same direction, right? Symporters means both of them are moving in the same direction. Because of this, so the higher pH in the apoplast is going to help. The high, sorry, it's lower pH. Hydrogen ion concentration is higher, so it's not higher pH. Because hydrogen ions are getting pumped out from the cell into the apoplast, so lower pH or higher acidic environment, right? That helps. So that is setting up that proton gradient, right? And then the sulfate ion, the phosphate ion, they're riding the proton gradient. Once the sulfate ion gets in, then it has to be absorbed into cysteine, right? Because that's the amino acid that uses sulfur. Phosphate, it'll be converted into like ATP and all these other things, right? They need to use that phosphate ion. Okay. So let's stop here for today, right? Yeah, yeah. I just wanted to mention one small thing. I'll talk about it next time. So I think we're already running out of time. Right? Are there any questions?